Of questions about that. So let me know if you guys are having issues. I actually just changed something in how I was compressing the file. So please let me know if there's an issue with, I guess I did the first one I did was lecture 8 because I was having issues with that. So if you guys have any issues watching lecture 8, please let me know. Okay. So it's exciting because it's very clear what we're going to be doing the next few days, and we'll very much be on target for our exam on Friday. So by now, wow, there's no one here. There are 75 people in this class. So you guys all get an A. The others? Yay. If only I could just hand out grades like that. Okay. So by now you guys should have all gone through the material. Has everyone at least taken a one-shot like glance? Have people not even thought about the exam yet? It's okay. I'm a last-minute kind of girl myself. But you really should this weekend take some time to go through the lectures and start generating a list of things you don't understand. Because then you have a week to figure those things out. Okay? And start drawing out all the processes, kind of like we do in recitation. Because then you'll know you understand it. And as I said a million times, don't study alone. Because if it's not clear by yourself, it's not going to get any clearer by yourself. Okay, so, and then this gives you all of next week to come see me to explain things. I've had a number of people come by with a whole list of things that were confusing, and we went through it together, and most people leave smiling. No one's left crying yet. So that's good. And if my office hours don't work for you, please email me. I'm not having many people take advantage of office hours. And it might be because you guys are like, I'm in class from 9:30 to 10:30. Well, that's fine, but I'm here. I'm not only here when I'm in front of you. I'm here all week, every day. Okay? Not to say you should stop by, but you should email me about when you should stop by. Okay. There was one entry for the cell dance contest. Thank you, people. I see people. I saw someone. Thank you for giving it a shot. I appreciated the effort, so it was 2 people who worked together, and they both got some extra credit points. Okay. So this is what we've done so far. A summary of the things that you should be concentrating on for the exam. Right, gene expression, as in what happens when you what kind of things need to happen so you can either have expression of a gene or not. Right, we spoke about that, and then what happens when you have a protein. The first place that we're covering is the secretory pathway, so understanding how it gets to the ER, what happens in the ER, ERAD, how does it get to the Golgi, what happens in the Golgi. And then where does it go? We talked about the lysosome. And we also spoke about coming from the plasma membrane to the lysosome. So there are 2 different ways to get to the lysosome for completely different purposes. So today we're finishing that bit up about endocytosis and talking about secretion. And then next week we'll do nuclear import-export and mitochondrial import. And then that'll be where we finish. So if you guys are wanting to get a head start on studying, there's no reason you shouldn't be able to, because this quiz this weekend will be focusing on the last bit of stuff that we are covering before the exam. So you shouldn't have that feeling on Wednesday of like, oh my God, she just taught this today and I have one day to learn it. You have actually a week. Yes?
Will you make the previous quizzes available?
The previous quizzes will all be open as of today when I post this weekend's quiz. There is a quiz this weekend, which will focus on nuclear import-export and mitochondrial import. So you guys feel free to get started on that. Yes?
And the nuclear import-export is going to be on the exam?
Yes.
Okay.
Everything is on the exam in this. Basically, if you look on the syllabus, it's divided into units. So the whole protein transport, that's everything under that is on the exam. Right up until 11:50 on Wednesday. Okay, if I don't cover it by the end of lecture on Wednesday, it will not be on the exam. Okay, enough about that. What were we talking about? We were talking about endocytosis, right? and we were talking about how receptors are recruited into clathrin-coated pits for endocytosis, and you should always be thinking when you're recruiting something into some vesicle, regardless of where it's going, there's some kind of control over that, right? It's not completely random, it's regulated. It's for a reason, for a purpose. So you should think to yourself, well, what's going to, you have a cell covered in receptors. What's going to make one receptor be endocytosed and another one just sit up there, right? So anyway, so there's an endocytosis signal, which is actually on the cytoplasmic domain of the receptor, and it's got the consensus sequence YXX, this thing. Which letter is that? Mu? I don't know. Sorry, I don't know I don't know. That one. Looks like a pitchfork. And that stands for any hydrophobic amino acid. And it's like all of these signals where it mediates some kind of interaction. In this case, it mediates interaction with, I believe, with adaptin, right? That makes sense. Okay. And the LDL receptor is slightly different from this consensus sequence. It has a different signal sequence, which is NPVY. But it's just important to understand the basic concept that it's regulated, and there's, we'll talk a minute about the regulation, and there's an actual binding site between the tail, right? The tail of the receptor, there's that tail, and adaptin, right? Okay, so I'm telling you there's a consensus sequence that mediates interaction with adaptin, so you should be thinking about this in the sense that there are many different receptors that are endocytosed, right? It's not just the LDL receptor. That was just an example I gave you. There's tons and tons of receptors, and they can accumulate in clathrin-coated pits. And why is this? They're all different proteins, right? So you have to be able to connect that with what I just told you about this consensus sequence for endocytosis. Okay, so what can you tell me? What's the purpose of the consensus sequence, this endocytosis signal? There it is, right? What's the purpose of that, and how does that relate to the ability to endocytose many kinds of receptors? Is my question clear, or you just don't know the answer? Can I rephrase it? So the way I might rate it on an exam would be something like this sequence is an endocytosis sequence. What does this say about the ability of receptors to accumulate? I don't know. That doesn't work well. Okay, so a lot of different receptors. That's why it takes a long time to write an exam. You have to phrase it right. A lot of receptors could be endocytosed in clathrin-coated pits, right? Not just the LDL receptor, many receptors, right? There's other receptors on the cell surface, transferrin receptor. And they could all be endocytosed into clathrin-coated pits. And we talk about how they're endocytosed by interacting with adaptin and clathrin, and then you get the budding in and all that. So how is this regulated? What does this have to do with this endocytosis signal I told you about? Yes?
Would the signal be the part that interacts with adaptin?
Right, so how does that make it easy for many types of receptors to accumulate in clathrin-coated pits?
It's kind of like a common adapter sort of thing where they can all hook up to the same piece.
Right, so adaptin's always the same, and I'm telling you this signal's always the same, except for in this one case. So if that signal's always the same and that's the binding site, that's how many receptors can accumulate in clathrin-coated pits and be endocytosed. Does that make sense to people? I see some nods. So shake your head like this if you don't understand me. Okay, good. Thank you. So the point I'm trying to get across is that there are a lot of receptors, and a lot of receptors participate in endocytosis because they need to bring things in from outside the cell, right? And they bring things in by accumulating in these clathrin-coated pits by interacting with adaptin. Okay? Right there. See right there? So there's this signal that's actually in the protein sequence of the cytoplasmic tail of the receptor that is the binding site for adaptin. It's on every single receptor that's going to be endocytosed through this mechanism. So that's why you can have 10 different receptors be endocytosed in clathrin-coated pits, because they all share that one sequence in their cytoplasmic tail. That's only 4 amino acids, right? Okay. This is a nice question for an exam, I think. Okay, so think about it, talk to your friends, you know. Okay, so I say it's regulated. Did I see a hand up? No? Okay. So I'm telling you it's regulated, right? Because receptors, so I gave you the example the other day of the LDL receptor, which does constantly keep endocytosing whether or not it's bound to any sort of ligand. But a lot of receptors are only endocytosed when they need to come in for some reason. Either they're bringing something into the cell or they're being downregulated. What does downregulated mean? Reduced expression, being made less, going away. Here my favorite protein, ubiquitin, is involved again. Okay, so remember we talked about ubiquitin, polyubiquitin, tagged proteins to be degraded in the proteasome. Well, here's a completely different function of ubiquitin. Here, monoubiquitin is added, so one ubiquitin is added to the cytoplasmic tail of a receptor, and that helps it to accumulate in the clathrin-coated pits. So ubiquitination is what regulates endocytosis. Completely independent of the proteasome, because where are these receptors going to go once they're endocytosed? Where? I hear whispers that say "om." Endosome and then lysosome sometimes. So if they're going to be degraded at all, it will not be in the proteasome, it'll be in the lysosome. And sometimes they go in the endosome, release their ligand, and then are recycled back up to the plasma membrane. And so that's kind of what they're showing you here, right? You've got your receptor, binds a ligand, there's some kind of signal kind of thing that recruits a ubiquitin ligase here and then adds ubiquitin, and then can interact with all the stuff in the clathrin-coated pit. So you have the clathrin-coated pit, endocytosis, and then you've got fusion with the endosome. So sometimes it goes, frequently, almost always, goes to this MVB pathway, which we'll talk about in a minute, multivesicular body. And then we'll so before it does that, it can either go back up and recycle, so you have this recycling pathway, or it can go to this multivesicular body endosome, which then will fuse with the lysosome and degrade whatever's inside. Yes?
So how does the monoubiquitin help? Like
It helps the receptor to go into the clathrin-coated pits. Yeah, because it needs to get into those clathrin-coated pits to be endocytosed.
So if it's not monoubiquitinated, it doesn't go in there?
Right. Now, not all the time. This is one general mechanism I'm teaching you, but it's never all the time everything. Yes?
Is that in addition to the adaptin being mediated, or is that?
Well, the adaptin is just what's going to bind to the tail
Right.
and allow recruitment of clathrin in the clathrin-coated pits, right?
Mm-hmm.
But there's no regulation there. That's just saying that adaptin's binding to the tail of the receptor. So the question is, how does this fit in with what I just went on about with adaptin and that signal in the cytoplasmic tail? That's just the binding site. Adaptin binds to the cytoplasmic tail of the receptor, recruits clathrin, endocytosis. But you need the receptor to get to the right place and for the adaptin and everything to actually interact. So that's a regulated process that's controlled by monoubiquitin. Did I see another? Yes.
How does this fit in with the
does it not? It doesn't at all. It's absolutely different. So that's why I keep talking about, thank you. So I keep talking about how you can get to the lysosome through two different ways. One is you're working at the lysosome. And you are an employee of the lysosome, and then you're coming from the Golgi, right? Because what are you? You're an acid hydrolase, and you work at the lysosome, and your job is to degrade things. That's mannose-6-phosphate and targeting. That's how those acid hydrolases. That's the bus ticket to your work. But when you're on the plasma membrane, that's different. That's like someone who's coming to visit the lysosome to. It's like they're going there to get a facial or something. I don't know. They're going there to be degraded.
Are there always just clathrin-coated pits, or are they recruited?
So in all cases, clathrin-coated pits are only made when they're needed to make a vesicle and bring something there. Like it's not just they're always there. It's a very regulated process, and that's why in this case, in the case of endocytosis, I'm bringing up ubiquitin because tagging the cytoplasmic tail of the protein is what basically says, hey, come on, adaptin, clathrin, and other proteins that are involved in endocytosis. Okay? Yes?
This probably isn't really important, but when you say there's recruitment of receptors, does that mean the receptors like move about the plasma membrane? Or?
Yes, the receptors move about the plasma membrane and kind of concentrate in an area that will become a clathrin-coated pit. Yeah. Yes.
So the clathrin-coated pits, is there like a common size where they all start to form and invaginate, or is it something that happens, they can be like many different sizes?
Oh, is there a certain size to clathrin-coated pits? Yes, you're a numbers guy, aren't you? I like this question. Actually, this is a math question in a way, because you have to think about the size of the protein. You have to think about the tensile strength of the membrane in a way, right? And actually, lipid rafts are areas of membrane that have higher levels of cholesterol, I believe, and cholesterol, I believe, makes a membrane more fluid, which makes it more easily bendable, and I believe receptors tend to congregate there and interact together in some way. So I don't know, like what you would have to do is really think about the cholesterol content of the membrane, so how flexible it is, and then it's a size issue, length of membrane versus number of clathrin, adaptin complexes that can fit and bend in a way that won't cause too much stress on the membrane.
So it might vary depending on what receptor is being endocytosed?
I'd say it varies based on receptor and based on membrane composition. But I don't know. I have no idea. That's me. This is how you can get through an exam, by the way. If you can give a logical answer that flows, then why could it be wrong? It could be right? That seemed who believed that? Who believed that what I said? That's what you learn in graduate school. You learn how to talk until someone believes you. Yes. Oh, wait. Start again.
Multiple receptors
Mm-hmm.
in the same part where the vesicle is being formed. Is it possible that something binding to one receptor will affect the binding of another thing to another?
So are you thinking that like if there's some kind of inhibitor somewhere, it might maybe inhibit the whole process? So the question is, if there are multiple receptors being drawn into clathrin-coated pits, could something inhibit the process, or could something affect the overall process because one thing will bind to one part and maybe affect something, right? I'm not sure. If I were a virus, that might be a good mechanism to mess around with something, but I want to say that the whole point of the ubiquitination is to kind of regulate what gets put into the clathrin-coated pits. So it's almost like you're selecting what goes in versus needing to leave something out, exclude something. You know what I'm saying? and it doesn't seem very efficient. And like what you're suggesting almost sounds like a haphazard way to endocytose. You know what I'm saying? Which is why I say it would be a nice mechanism for a virus to mess around with things. Because if I were a virus and I wanted to prevent, let's say I wanted to prevent EGF signaling, some kind of signaling, and I wanted to prevent endocytosis, and I know the whole ubiquitin thing is going to happen because that's part of the cellular process, but I want to prevent that, I might make a protein that'll go bind to the cytoplasmic tail and prevent adaptin binding, right? I don't know. I bet you we could look it up and find some. Ooh, let's make an extra credit. I'm looking at you because you gave me the signal last time. Okay, let's make an extra credit. Is everyone ready?
No.
Is there any known example of a pathogen inhibiting endocytosis? And what is that? And what mechanism does that pathogen, virus, whatever, use? And does it have any effect on interaction of the receptor? That can't be us. Does it have any effect on the interaction of the receptor with adaptin and clathrin? It is for life on YouTube. I'll say it again. Is there any known example of a pathogen interfering with endocytosis by somehow interacting with cytoplasmic tail adaptin, clathrin, something like that, and preventing clathrin-coated pit formation and endocytosis? That means you need to find a paper where someone studied some virus or some pathogen, and they show that that virus has a protein that inhibits endocytosis and how. And you can't just send me the paper. You need to explain what they show. Question in the corner?
Yeah, just the last sentence. You're preventing something.
Endocytosis through clathrin-coated pit. Because I like that. I like that question because it shows me that you're thinking, which is important for success in this class. It's important to think. If you don't think, bad things happen. Okay. Okay, so let's move on. Oh, let's move on. Okay, so here's a practice problem for you guys to chat with your friends about. You're studying a new receptor. You're trying to determine whether it participates in receptor-mediated endocytosis through clathrin-coated pits. You know the protein's amino acid sequence. How can you use recombinant DNA technology to address these research questions? So basically, is it a receptor that participates in receptor-mediated endocytosis? What would you look for? What would you do? How would you prove your point? So chat with your friends for like 2 minutes, and we'll reconvene. He never closed this.
Well, since you know the amino acids, you just figure out
But that one's like right down there.
I totally saw the girl who sits next to you. Yeah, leaving as I was coming here, and I was like, "You're going the wrong way," and she giggled. And so I was like, "Well, maybe she's just like going against me," but actually, so she never came. She was probably like, "Uh oh." Dr. Ehrlich saw me skipping her class. Or I wonder if she was the one who told me she had something going on. She probably did. I know. She told me. There's so many people who tell me so many things, and I just am like, well, I don't really care. Just come to recitation. So. Okay, guys. So what would you do? I want to hear from someone who never says anything and is terrified that I'll judge them horribly. That way I can judge you horribly. You always say something. But I bet you have something good in there, so I'll get to you after I talk to you who doesn't say something. Okay, so look for that consensus sequence. Okay.
I think that was what I had.
Okay.
The white explorer.
Yep. Yep.
for the drawing.
The pitchfork. Yep. Look for the consensus sequence, and what would you do to it? Would you so you see it there, but are you so if it participates in receptor mediated endocytosis, what can you look at, and what can you do to show that that is indeed a consensus sequence for endocytosis? Someone else I never see. Yeah. Right, so you can remove it by mutating it or cutting it out or something like that. And what would you look for? What would you expect to see if that sequence was gone? No endocytosis? Okay, so how would you look to see if something was endocytosed? What would you just based on what I've taught you, you know what endocytose receptors interact with, you know where endocytose things go. Yes.
If it's not at the cell surface, like at the membrane, you would know that it wasn't endocytosed, or it will still be inside of the.
Oh wait, okay, so if it's not at the plasma membrane, will it be endocytosed or not? Endocytosis brings things in from the plasma membrane.
Oh.
So if it's not there anymore, was it endocytosed? Yes.
Yes.
So let's go the opposite of that. Okay, what were you gonna say?
Maybe like so you could tag it and then see like maybe with a dye and see if it's gonna end up in the lysosome.
Okay, right. So you can tag it. You could do that with GFP. Make it break green receptor, and see if it goes to the lysosome. Very good. That's very good. Another one. I saw your hand up. No. Anyone else?
I was just gonna say tag the receptor, tag the cell wall.
The plasma membrane. Yep, you love plants and bacteria. Yes, tag the receptor and tag the plasma membrane. So see if it localizes to the plasma membrane and goes away. Actually, there's a very cool experiment called FRAP, I believe, which might be good. FRET. No, that's a different one. Well, either way, it's fluorescence loss after photobleaching. Yep, there you go. And where actually you can look at your plasma membrane and have a fluorescent protein, and you bleach it to see if it comes back. That would actually be better for sending something up to the plasma membrane. See if it goes away and comes back. Good for recycling. Anyways, sorry. I digress. Okay, what else can you do? So let's say you have no microscope and you can't look at anything, and you have to look at protein interactions. Yes.
Can you just bind mononucleotide into it and see whether it's recruited into the plasma?
Okay, so you're bringing up ubiquitin, and that's a great idea because that's something no one has mentioned yet. So you would need to do what with ubiquitin? If you bind it to it, then does it tell you anything about, I know what you're trying to say, see if it binds monoubiquitin?
Yeah, you said that monoubiquitin kind of recruits it into the receptor, into the plasma
Yep.
codes. So if it binds and it recruits it into the plasma codes, and then it's gonna go through enzyme.
Right. Okay. So basically, then we comes down to how you do that, right? So exactly. So you would look to see, is the receptor being monoubiquitinated, and is the receptor, if you mutate with the lysine where the ubiquitin's added, is it endocytosed or not? Right. So that's something else you can do. Okay. So I think that's good. And so there was one other thing that no one said, and that's just basic interaction with adaptin, right? Because I told you that signal sequence interacts with adaptin, right? So you can actually just run a gel and look and you can immunoprecipitate a protein to see what it's interacting with. So you have your wild type receptor and your mutated receptor, and you pull them down, literally pull them down by attaching them to heavy agarose beads and using a centrifuge, and see what it binds to. Your wild type protein should be interacting with adaptin, right? If it's ubiquitinated, and your mutated protein, should it be bound to adaptin? No, right, because you mutated the binding site. Okay, let's move on. If you have questions about this practice problem, feel free to come see me either after recitation sometimes or office hours or talk to your friends. Okay, so I'm going too slow today, so I'm going to speed it up a little bit. So unless cargo is diverted elsewhere, the default pathway for molecules taken in through endocytosis is the lysosome. Okay, so basically you go to the lysosome unless you're diverted elsewhere. And what is that usual elsewhere? If you endocytose and you don't go to the lysosome, where might you be going? Back outside, right. So recycling back up to the plasma membrane. Right. So basically you can have recycling of the receptor to the plasma membrane, the same plasma membrane, in polarized cells where like there's a top and a true top and a bottom, like cells like endothelial cells or cells, skin cells that actually interact with other cells rather than, let's say, blood cells in the blood like white blood cells. You'll have transcytosis, which you can actually have transit of a receptor from the top to the bottom, the side to the top, and then also you can have degradation in the lysosome. So the LDL receptor, which we talked about the other day, was the example of recycling the receptor, right? It binds to LDL on the plasma membrane. You have endocytosis, fusion with the endosome, and in that you have two things that happen, right? You've got concentrating of the receptors in these kind of like tubule things that form in the endosome, and those bud off and become the recycling endosome that brings it back up to the plasma membrane, and then the rest of the endosome fuses with the lysosome and breaks everything down. So that's an example of recycling your receptor. So then there's another example of recycling your receptor, but this is another one because you're recycling your receptor and your ligand. And why would you want to do this? Well, an example of why you would want to do this is the transferrin receptor. What does transferrin bind? Transferrin, ferrous, iron, right? Transferrin. Binds iron, brings it into the cell. So this is an example of recycling both, because you'll have your transferrin receptor that has transferrin bound, and it's either in an iron bound state or a not iron bound state, and what's being released is the iron. What needs to stay bound is the transferrin on the receptor. So what you actually end up doing is you endocytose your transferrin receptor, which would be actually your ferrotransferrin when it's bound to iron, right? Because you want to bring in the iron. And then you go in your plasma-coated ped and your endosome, your late endosome, and then you have this pH drop, which results in release of your iron, right? You want to release the iron inside the cell. And then the apotransferrin lacking the iron goes into the recycling lysosome, endosome, which then goes back up to the cell surface. So that's almost exactly the same as LDL receptor, except for what's the one difference? Uh-oh.
The ligand is attached.
Right, the ligand stays attached. Sometimes my questions are very simple. I'm not trying to trick you. I'm trying to make you verify what I just said. Yeah. So the difference is in LDL receptor, only the receptor goes back up. LDL stays in the cell, right? And in transferrin receptor, the transferrin goes back up along with the receptor. So why is this an economical way for the cell to take up iron? I always like to talk about economy in the cell. You want to be economical in how you run your business. If you're a cell, yes.
You don't have to keep making the ligand and the receptor.
You don't have to keep making the ligand and the receptor. So in the case of the LDL receptor, well, LDL is out floating around in the bloodstream, so it's a different story, right? Here, and there, that's why recycling the receptor is useful, because you don't have to keep making more receptors. If you keep degrading things and making more, isn't that a huge waste of energy for the cell, right? So in this case, it's efficient because you don't have to keep making transferrin as well as the receptor, right? And it's just going to go up there, bind a molecule, and bring it back down, right? Okay, do you guys see that? Good. Okay, let's move on. So then we'll talk about, for a second, receptor downregulation through endocytosis. So now I want you to remember this concept when we go to signaling, because signaling is all about something binding to a receptor and sending a signal to the nucleus, which regulates gene expression, right? So if you're going to downregulate your receptor, what are you going to do to your signaling? Is it on or off? Off. And what's going to happen to gene expression if you turn your signaling off? It's going to be off. So that's how you turn things off. Why would you need to turn gene expression off? If you don't need something, right? Okay, just big picture. Huge picture. Don't express genes you don't need, right? Don't make proteins you don't want, right? Okay, so this is one example that I just kind of like the way this figure looked. It's very basic. You've got your ligand that binds to your receptor, and then you've got there's extra proteins in here because this is from a paper, so don't worry too much about it. Then you get downregulation in your clathrin-coated pit, and sorting to the endosome, and it could go to the recycling endosome if it's needed, but if it's not needed anymore, you can shut it off by sending your receptor to the lysosome. And more specifically, that's a very simple way of thinking about it, but usually receptors are downregulated by sequestering them in multivesicular bodies. And so what does that mean? So you're just going to have to follow me here for a minute, right? So you've got your receptor, and it binds to its ligand, and it's ubiquitinated and endocytosed. So I want you to notice you've got your receptor ligand binding part up top here on the outside, and then you've got your cytoplasmic domain, the signaling part on the inside. So when you endocytose and you're in a vesicle, your cytoplasmic domain is still in the cytoplasm, right? Right? Okay, so if anyone can think about signaling for a minute, which I haven't taught you much of, but most of you know that signaling usually involves a cytoplasmic tail of some receptor being bound and phosphorylated by things, which then bind and phosphorylate other things, and all the way down to the nucleus, where some transcription factor goes into the nucleus and activates gene expression in some way. That's how signaling works. Did everyone know that? Does everyone know that now? Okay. Well, so I just said there's a cytoplasmic tail that's binding to things in the cytoplasm. So when you endocytose, that cytoplasmic tail is still in the cytoplasm, still able to signal. But isn't the purpose of this to stop the signaling? Right? So you need to sequester that cytoplasmic tail from all the signaling proteins that live in the cytoplasm. Right? So what you do is you actually bud into your endosome one more time. So you see it here, and you actually have extra invagination, which then results in your receptor being completely inside what's called a multivesicular body. It's exactly what it sounds like, because it's multivesicular body. And here it can't signal anymore. And then it will go to the lysosome and degrade everything. Yep. questions about that concept. It's an important concept to understand. It's pretty simple if you understand signaling, but if you have no idea what I mean by signaling, it's going to completely elude you. Yes?
Is there like a case where they won't degrade them, they'll just like hang out in the multivesicular bodies, or are they always just taken right to the lysosome?
So HIV actually is a great example of budding into multivesicular bodies and not being degraded, but it being like a factory for making virus. So yes, there are examples of that happening, and sometimes I'll have to think a little bit more about a non-virus example, but a lot of viruses do bud into multivesicular bodies and then mess around with the machinery to then direct it towards the plasma membrane, and then it's like this huge release of virus out of the cell. Like imagine a multivesicular body full of virus that then fuses with the plasma membrane. Lots of virus. Trying to think if there's an example. I feel like I've read things in the past. Give me a little bit and I'll try to get back to you on that one. Okay. So these questions are good for you guys to think about what you're studying. What's the purpose of sequestration in a multivesicular body? I just told you that. Why? To stop the signaling, right? Because you want that cytoplasmic tail inside the multivesicular body. Why not just send the receptor complex directly to the lysosome in an endosome? Well, then your cytoplasmic tail will be signaling all the way down. So if you want to shut something off, shut something off. Don't be wishy-washy about it, right? Cell's not very wishy-washy about things. Okay, questions about this before I move on? Good. Okay. Oh, we're almost there. We've got about 5-6 minutes. Endosomes can store membrane proteins for use when needed. So an example is these glucose transporters. What do? They transport glucose. They transport glucose in response to insulin signaling. Makes sense, right? Blood sugar goes up, insulin binds to insulin receptor, sends a message to the cell to send these glucose transporters to the plasma membrane so they can bring in glucose, lower blood sugar. So that's how it works. So they actually kind of hang out in specialized recycling endosomes, kind of near the plasma membrane. And when insulin signals, there's signaling that happens that does a whole lot of things. But one of the things that signaling does is it says, hey, glucose transporters, time to head up to the membrane and bring in that glucose into the cell. So that's another thing endosomes can do. So we talk about recycling endosomes and how they bring receptors back up. Well, sometimes they just don't quite get there and they hang out close by until a signal tells them to go. Okay? So what kinds of cells might have large intracellular pools of glucose transporters? Stomach cells, is that what I heard? Maybe. I'm thinking of 2 cells that use lots of glucose. Muscle. What else? What other cell loves to take in lots of glucose? Yes. Oh, brain, probably. You're probably right. I wasn't thinking of that one. Fat cells. Okay. Good. So let's talk about secretion for a minute, right? So we've been talking about, we were talking about things going out, right? For a long time, we were talking about proteins being processed and moving outwards, right? And then we shifted and we started talking about things coming inwards to the lysosome, right? So now we're going to go back again to things moving outwards, right? Because we went Golgi. From the Golgi, you can go to the lysosome. From the Golgi, you can go to the plasma membrane. From the Golgi, you can be secreted, right? And that's also called exocytosis. So 2 major concepts, constitutive versus regulated secretion. Very basic concepts, right? Constitutive means what?
All the time.
All the time, thank you. constitutive all the time, secreted all the time. There's no regulation. It's just protein's made. The only way it's regulated is whether or not that protein's made, right? Protein's made, goes through the ER, the Golgi, it ends up in, there's no transmembrane domain, right? So it's in the middle of this vesicle that buds off and goes out to the extracellular space. What does regulated mean? It's regulated. So a lot of things would be that makes sense that a lot of things will be regulated, right? I mean, a lot of things will be. I guess there's both, but with regulated secretion, there's usually some kind of receptor and some kind of signal, like some hormone or neurotransmitter or some immune-related molecule that will bind to a receptor and cause signaling. And these arrows are very basic. The signaling doesn't go right to there. Signaling usually does a lot more than that. But anyways, and that kind of tells the these vesicles that are packed full of whatever secreted protein to go fuse with the plasma membrane. So an example, we'll get to that in a minute. Okay, so the constitutive secretory pathway is the default pathway. If nothing redirects a protein, it's going to be secreted out of the cell. And we kind of talked about it. If you cut off the K-del from a protein, it's not going to go back to the ER. It won't have any other kind of tag on it, and it will just be secreted out of the cell, right? So you want to think about, okay, so something in the Golgi, what can happen to it? What determines what happens to it? You can have diversion to lysosomes. What diverts something to a lysosome? Mannose-6-phosphate, right? And then you can have diversion to a regulated secretory vesicle. And I haven't told you what that mechanism is, but maybe you can guess. There's something that keeps it in. I don't know. I don't think I'm even going to teach that to you because I don't know if it's completely known, actually. But you can guess it probably there's some kind of sequence that targets it into a certain kind of vesicle versus another kind, right? There's probably some kind of receptor involved, kind of like M6P receptor, K-del receptor. It's all the same theme, right? And then if there's nothing to send it one way or the other, it's going to be secreted out of the cell. What about plasma membrane proteins? What's going to happen to them? How come it's not secreted out of the cell? You need it, but what within the protein determines whether it's going to be at the membrane or secreted out of the cell? Let's see if I see any new hands. Okay.
I guess just the way that it folds with the hydrophobic and hydrophobic regions.
Has some hydrophobic regions. Where are those hydrophobic regions? In the membrane. Can it be secreted if it's stuck in a membrane? No. Okay. Okay, what's missing from this figure? There's one thing. So it's showing this flow from ER to Golgi to lysosome, secreted, secretory vesicle. What's missing? There's something big missing. Yes?
Vesicles going back to the ER.
Right. Vesicles going back to the ER because don't forget there's all these proteins that accidentally go that way and need to be sent back to the ER, like those ER resident proteins, right? Absolutely. Okay. So secretory vesicles they contain specific things that are secreted in response to a signal. So there's a receptor, there's ligand, there's signaling, and that results in mature secretory vesicles containing lots of some kind of molecule. So, so what kind of cells might have lots of secretory vesicles? Think about cells that secrete tons and tons of things.
Intestinal cells.
Intestinal cells, like mucus and stuff like that. Anything in the immune system?
Endocrine glands.
Endocrine glands, hormones, so things that secrete hormones. Yep. Anything else? So you guys are thinking, and those are all going to be regulated, right? You don't want to be mounting an immune response when there's nothing there. Maybe the mucus not so regulated. That's probably constitutive, right? Because your intestine needs, I don't know, maybe I don't know. We need to think more about that one. Okay, so this is just showing how it works with insulin, which we just talked about anyways. There was one thing, oh man. Okay, we're not going to make it all the way through. We will finish this next week.